Okay, this is how visual information is traveled through the cranial nerve number 2, and also how it finishes in the visual cortex and what happens after that. So this diagram is showing you the central visual pathway. Central visual pathway. So let's say, for example, we are looking at the orange wall on the left and blue wall on the right. I should say green wall on the right. And this information will travel through the pupil and retina. So retina contains the photoreceptors, and one thing you realize is when it goes through the pupil, the light is inverted, which means instead of that being left and that being right, which is like orange and green, it's reversed. So now the green visual information is on the left side of the retina, and also the orange visual information is on the right side of the retina. So the visual information is inversed in the retinal membrane. So its left and right is inversed, also up and down is inversed, but we only are looking at the left and right here. So let me just say this is the image from the right visual field, and this is the image from the left visual field. Same with the right eye, right visual field in the green, and left visual field, which was orange wall. So that's inverted, and this information will travel through the cranial nerve number 2, which is the optic nerve. So optic nerve is this part. Actually there's a labeling on the left side, so I'll just link that with this. So this axon is our optic nerve, cranial nerve number 2. And in the optic nerve, near the in front of the basal brainstem, there's a chiasm, which is the decussation of an axon of the optic nerve, and only the information from the nasal retina decussates to the other side. So let's have a look at the nasal retina and temporal retina. So if we divide the retina into two, you'll see on the left and right side of the eye, left eye, this side, which is close to the temporal bone, that's the temporal retina, and the other half, which is medially located, which is close to the nose, this is the nose, that's our medial retina. Same with the right eye, medial retina close to the nose, and temporal retina close to the temporal bone. And it is the nasal retina, so information from this side and information from that side will cross over to the other side. And temporal retina is ipsilateral, which means it doesn't cross over, it stays in the same side. So this is only shown for the right visual field. So the right visual field is temporal retina for this left eye, and the right visual field is left, which is the medial nasal retina. So sorry, this is nasal, not M, so nasal retina. So nasal retina decussates, so you can mark this symbol decussation here. And I didn't draw the orange visual field, which is left visual field, but you can imagine the axon for the nasal retina on the left eye will cross over to the other side, but not the temporal retina. So this is for the orange visual field. This is you can practice this with the other template, which is in the tutorial, back of the tutorial manual. So the nasal retina decussates and temporal retina stays in the same side, and as a result all the right visual field information travels through the left side of the tract, and this tract after the optic chiasm is called optic tract. This is the optic tract, and that's the point where nasal retina decussates. Okay, just give some description of the nasal retina here, close to nose. If we split the retina into half, so the medial side is the nasal and close to temporal bone, which is the lateral side, that's the temporal retina. After the optic tract, some axon can go into the midbrain, so this is the bifurcation of the optic tract, and one can go to the midbrain. This area is called pretectal area, which we have covered in week 6 for pupillary light reflex. So this is the one of the part of the afferent optic nerve that links the afferent limb of the pupillary light reflex. So that's responsible for pupillary light reflex. That's where you can find the interneurons, which goes to the ocular motor nuclei. And the other function for pretectal area is it's a site for optokinetic reflex, which we will cover in the next slide. So optokinetic reflex is related to pretectal, associated with pretectal area. The other branch can go to the superior colliculus. So this is the superior colliculus. And when it goes to the superior colliculus, it can initiate the neck movement through a tectospinal tract. So this tectospinal tract not only receives the information from vestibular nuclei, but also receives information straight from the visual information. And also it can initiate a type of eye movement called saccades, which we will cover in the next slide. So that's the branch that goes to the midbrain part, and the major branch goes to the thalamus. And again, thalamus acts as a relay center for the sensory information, and this particular area or nucleus of the thalamus is called lateral geniculate nucleus. Lateral geniculate nucleus of the thalamus. And after the thalamus, it goes to the primary visual cortex, which is located medially in the hemisphere. So this is the primary visual cortex, and as all the axons kind of radiates towards the primary visual cortex, this axon is called optic radiation. And you can label this part of the cortex as our primary visual cortex near the calcarine sulcus. And when the information arrives at the cortex, we can be aware of the shape, size, or color or texture of the object. So we can discriminate those aspects of the visual information. And all the visual information will be related to the opposite side of the visual field. So you can see all the green information ends up in the opposite side, which is the left visual cortex. and you can practice drawing the right side of the left side of the visual field ends up in the right side of the primary visual cortex by doing this the opposite way with this drawing. So that's our central visual pathway. And what happens after that? We can process the visual information further to get the meaning of the visual information or recognition of the object we're seeing. So medially there's a visual information, but that visual information can go to association area of the visual cortex. So there's two parts of association cortex, visual association cortex, and one is called dorsal stream, which goes to the dorsal part. Remember, this is the dorsal and that's the ventral for this axis, because of the flexure of the cervical flexure of the nerve system for the fetus. So this is the dorsal stream, and that's the ventral stream. So dorsal stream is also known as action stream, and the function of this stream is after discriminating the object, you can also analyze the motion of the object or movement of the object. And if a person damages this stream, for example by stroke or a traumatic brain injury, one can have this symptom called motion blindness. So the patients suffering from this cannot analyze emotion at all, and what they perceive around them is all the still pictures like this. When you're pouring the water in the glass, she or he will just see this picture in the series and can't recognize the motion. So mostly they're really afraid of going out, because if you imagine, like, say the one of the description here, people were suddenly here or there, but I have not seen them moving. So it's very dangerous for them to be in, for example, on the street where the car was suddenly in front of them, and you can't really estimate the motion of the car, and they have to rely on the other senses, such as auditory senses or tactile senses, to guide or predict the movement of the object around them. So that's kind of stark example of the function, showing the function of this action stream or dorsal stream. On the opposite side, the ventral stream, this is also known as perception stream, and they can recognize the motion because the action stream is intact, but if they damage this part of the visual association cortex, they have trouble recognizing or perceiving the object. So analysis of the object, recognizing, oh, this is pen, or oh, this is glass, and so forth. This particular area which processes the face of the person, and if you damage that particular part in the ventral stream, you can suffer from prosopagnosia, which is the face blindness. And they can recognize someone's nose, mouth, eyes, so they have no trouble naming those parts, but they can't really organize that as a whole and recognize, oh, that's my mom, oh, that's my dad, and so forth. So for them, it's one of the descriptors for their symptom is every face looks just like pebble, and you can't really differentiate because all pebbles look the same. It's really hard for them to differentiate, oh, this pebble is John, and this pebble is Tom, this pebble is Jane, and so forth. And another stark example of this is the patients reported this: at the club I saw someone strange staring at me. So he asked the staff who it was, and it was actually himself in the mirror. So he was looking at himself in the mirror, but he didn't recognize his face, and he thought it was some other person staring at him. So this is the stark example of face blindness. And those are the two major functions of the visual association area. Okay, let's move on to eye movements so that we can actually link the vestibular system to visual system later on. We have 5 different types of eye movement, but broadly we have 2 major functions for eye movement. One is gaze stability, and the other one is directing the gaze. In order to stabilize the gaze, well, why would we have to stabilize the gaze? Let me just give you an example of how dizzy it is if you don't stabilize your gaze. So this is an example, the video clip. The camera is actually moving with my head, but because I'm walking, every step I make, it actually shakes the paper and can't really stabilize the camera, because my head is moving with the entire body. So it's really dizzy. You will really feel dizzy if you don't really have this stabilizing mechanisms of the gaze. So there's 2 types of stability for gaze stability. One is vestibuloocular reflex, and that's particularly made for fast head movement. There's another type of reflex which was associated with pretectal area, optokinetic reflex, that's responsible for slow head movement or movement of the large object in the visual field. So there's 2 types to stabilize the gaze, and we have 3 types of eye movement to direct the gaze. Saccades, it can be reflexive or voluntary, but saccades means fast eye movement or bringing new object into central vision and hold it there, hold the gaze in the spot. There's another type of directing gaze, smooth pursuits, and this is following a moving object. So if you follow my cursor, then you're using the smooth eye movement, smooth pursuit movement, eye movement. So this cursor is moving left to right, and that's your smooth pursuit eye movement. But if I say, look at the picture, and you direct your gaze at the picture and hold it there, that's your saccades. And especially that's for voluntary saccades, because you voluntarily moved your eye to one particular spot. And the 5th category of eye movement is vergence, and it's the bringing the eyes towards or away from the midline. So vergence is to accommodate or focus near and far objects. So that's vergence, which was number 5. And the other movement, all movements, are conjugate movements, which means both eyes moving in the same direction, right this way. So it's representing the eye movement 1 to 4, vestibuloocular reflex, optokinetic reflex, saccades, and smooth pursuits. They are all conjugate movement, and we have vergence. So let's have a look at gaze stabilization, which is the first one is vestibuloocular reflex, VOR in short. And this reflex has short latency, about 5 milliseconds. which means it reacts really fast to perturbation and can accommodate high frequency head movements, such as the head movements derived from the locomotion, especially in the heel strike. There's really high frequency components in the heel strike, and this VOR can compensate and stabilize the gaze. So this VOR is responsible for us to read the books while walking. So you can try this and you realize you can actually do this. It's not as dizzy as you think. It's different from camera because we have this reflex mechanisms. So how does it work? First we have head rotation or head perturbation, and let's say we activated the semicircular canal, so it had rotated in the particular, let's say this, use this example for instance. So this is the leftward rotation of the head, and this information goes to the vestibular nuclei, and through the MLF, which we covered as one of the output of the vestibular nuclei, which connects to the cranial nerve number 3, 4, and 6, it can control the eye movement in relation to the head movement. So this MLF is very important. Let's spell this out: medial longitudinal fasciculus. Fasciculus. SI. Sorry, it's SC. And the pathway of the vestibuloocular reflex is simplified in the following diagram here. So the left head movement will stimulate the horizontal canal on the left side, so it will generate the action potential, and that's the vestibular nuclei. So vestibular nucleus is shown there. It's synapses with, so there's a cell body, should be a cell body, and synapses with, this is the abducens nuclei, and also further synapses with the ocular motor nuclei, and this axon is our MLF. So that links the vestibular nuclei to the cranial nuclei, and because the 6 and 3 nucleus is coupled in the opposite side of it, it actually activates, so it activates the abducens nucleus on the opposite side, which is the right eye, and it cross over to the other side, so it double crosses here, and it activates the left side of the oculomotor nucleus, which contracts the adductor medial rectus and lateral rectus for the right eye. And if you contract those muscles, the end results will be rotating the eyeball towards the right, and that way if you rotate the head in the left but your eye is rotating opposite way, you can stabilize the gaze, you can look ahead in the same direction as before rotating the head. What happens to the other opposite muscle is, now there's another connection here, and this time it's the inhibitory interneuron, and it automatically inhibits the left abducens nerve, which inhibits the contraction of the lateral rectus of the left eye, and also it crosses over to the other side with coupling, so it's inhibiting the medial rectus of the right eye. So this will be relaxed and this will be contracted. So the end result is the same. The eyeball will move, it will facilitate the eyeball to move in the opposite direction of the head movement. So let me just give you another demonstration of this VR demonstration. So your head is moving left and right, and the eyeballs are moving the opposite direction, and end result is the eyeball is directing straight. It's not really strictly straight, but you can just imagine the eyeball is looking straight. Yeah. Oops. So the opposite direction, counter rotation, helps the gaze to be stabilized. So it applies to not only the left and right, but also up and down as well. So in that way, we will be able to, we are able to read books while walking, because this subtle adjustment in the eye movement opposite to the head movement occurs really quickly. So this is for fast head movement. What about slow head movement, or if the bigger object is moving slowly in the visual field? In this time, it's using the vision to guide the stabilizing the eye movement, stabilize the gaze. And the reason for this is for the hair cell, I should say hair cell, or let's say vestibular apparatus receptors are fast adapting receptors, which means it's not sensitive to slow or prolonged rotation. So the signal from the receptors will be very small, and we can't really stabilize the gaze for slow movement because it will not really elicit any action potential for the cranial nerve number 8. So instead we have to rely on cranial nerve number 2, which is the retinal information or visual information. So we detect, instead of gaining the information from the vestibular apparatus, we gain information from the visual field, or visual through the visual optic nerve, sorry, and then stabilize our eye according to that. So for slow head movements, the pathways are very similar to VOR, but it goes to pretectal area, and from pretectal area in the midbrain goes to the vestibular nucleus, and through MLF it activates the 3, 4, and 6 in a similar way as the VOR. The only difference is when you look at this part is exactly the same, the way it activates the vestibular the cranial nerve number 3, 4, and 6, but the only difference is the source. One is coming from the vestibular apparatus, and the other one is coming from the pretectal area carrying the visual information. The implication for this is the vestibular nuclei have no way of differentiating whether that signal is coming from the vestibular apparatus or if it is from the vision. So it gets mixed up, which means you might have the experience of, like, you're sitting in a train and there's a window in the train, and outside the train, so you're sitting, let's say a person is sitting watching outside, it's no longer a person, outside, and there's another train that is moving this direction, and you feel this perception of your train is moving in the opposite direction, and your train is actually not moving, it's stationary, but you've actually perceived the motion because the other side, the train next to you, is moving. So this is actually activating, it's caused by activation of this optokinetic reflex, which is activated by the visual information. So this type of illusion is also known as vection. So illusion of the movement created by this optokinetic reflex. And this is due to the vestibular nuclei gets input from both semicircular canal or optolytic membrane and also the pretectal area, which carries the visual information. Let's see. Let's just stop here for a sec. Okay, let's move on to directing the gaze and we're going to look at saccades and smooth pursuit eye movement. So let's have a look at saccades. That's a French word for pull on the reins. So what it does is move the eye rapidly to the spot and hold it there. So if it can happen in involuntary fashion. So if I say look at the right side, look at the cursor, which is stationary, and you can use your saccades to direct your eye to your gaze to look at that cursor in the right top corner. So how this works is through this area called frontal visual field, which is near the primary motor cortex. So if this blue line, it presents the primary motor cortex. And it's anteriorly positioned around the homunculus of the eye area. So this is the homunculus. Let's say this is the homunculus of the eye area of the face, and the frontal eye field is located anteriorly to that particular homunculus. So this is frontal eye field. And that's where you can initiate the voluntary saccades. So I want to look at the right top corner, and you can move your eye there, and this is the initiation of that signal. So it goes to, so it starts from the frontal eye field, which is number 3, and goes to the superior colliculus, which is number 4. So let's label that the superior colliculus. And then after that it goes to reticular formation and to the cranial nerve 3, 4 and 6 to move the eye. And it has to go through reticular formation because it is a network that is responsible for calculating the right velocity and position of the eyeball to direct the gaze in the right direction. So 3, 4 and 5 is how we move our eye to direct our gaze voluntarily. There's different types of saccades, for example, related to attention. So it occurs consciously, so it's subconscious control and related to our attention. And we can see that pattern of the gaze using eye tracker. So this is the representing the gaze pattern. So if we look at this picture, we normally look at most of the time eye and nose and this area. So this is not occurring voluntarily, it's subconscious control, and this is shown in this circuit, which starts from the visual cortex, primary visual cortex, and process further in the post-parietal cortex, which is number 2. So it starts from 1, goes to 2, and goes to frontal eye field, and process further to superior colliculus, reticular formation, and cranial nerve 3, 4, and 6. So it's 1, 2, 5 that represents the post-parietal cortex, which is related to attention-related gaze control. The other example of this attention-related gaze control is the difference between male and female gaze pattern, just for your interest. So the red area indicates the high gaze area and fixation area, of the gaze, and that's subconsciously controlled. So you can see the stark difference between men and women and where they look, and that's related to this post-parietal pathway. And thirdly, reflexive saccades can occur, and this is directly from the superior colliculus. So from central visual pathway, the branch can go straight to the superior colliculus, and that can initiate the saccades. So this pathway is starting from the fore and through the reticular formation directly to the cranial nerve number 3 to 6, controlling those nerves. And this works when you see a fast moving object in your periphery, and you can automatically direct the gaze to the object, and that's reflexive saccades done by superior colliculus. So those three are the pathways for the saccades, 3 types of saccades: voluntary, subconscious related, attention related control, and thirdly reflexive control. The last type of gaze directing mechanism is related to smooth pursuit control of eye movement. So this diagram shows the smooth pursuit eye movement, and it has complex steps, so I don't really recommend to memorize all the steps behind it, but just to recognize it's more complicated than the saccades. And the reason behind this is the moving object, and tracking the moving object not only requires the calculation of the eye to direct the gaze in the right place, but also the head motion as well. So there's very complex calculation going on with this smooth pursuit control. So it's more circuitous pathway than that of the saccades. So where we start from the first primary visual cortex, and the next step is, rather than going to post-parietal, it goes to temporal eye field, which is number 2, and goes to frontal eye field, which is number 3. And it has to go to the cerebellum to do the complex calculations. So this is the cerebellum. And if you can recall from week 2, with the structure that links the big brain to the small brain was pons. So frontal eye field, the information from the frontal eye field has to go through pons. So for this diagram is pons. It goes to the cerebellum, and the output is calculated from the cerebellum, and the 5 represents the vestibular nuclei, and that information goes to the reticular formation, and finally to the cranial nuclei 3, 4, and 6. So that allows us to track the moving objects in space.